Okay, Duran Brown here. I'm your host, and I'm here with Diego Cordero.
Exactly. You got it.
Cordero Cervantes.
Perfect.
Okay, awesome. We actually went to the University of Utah with each other, and I'm just amazed at what he's been doing with his career. And I knew somebody I definitely had to get on the show. After attending University of Utah, Diego went to Princeton, I believe, and he was a research assistant at Princeton. And he later went to Cambridge in America and studied under Harvard. Is that correct?
That's right. Yeah.
Okay, one thing I do remember about Diego is that he was always full of positivity. Passion used to be his favorite word, passion, passion. And he was a hardcore boarder. Everywhere I saw him with a skateboard. So
True.
I love to see people leveling up in all areas of their life. But before we get too much into it, can you give a little bit about your background, where you're from, your interests growing up, and how you actually got into studying the brain?
Yeah, sure. Man, where to start? I was born and raised in Bolivia, in a city named La Paz, which is, if you've heard, one of the highest capitals in the world in altitude. We're at 4,000 meters above sea level, which is, I think, over 11 or 12,000 feet. So pretty high. Um, and unlike most places in South America, I actually grew up in the cold, right? We're in the mountains, so people always say, "Oh, you probably miss the hot weather in South America." And I'm like, "No, no, you don't understand. I come from the mountains."
Yeah.
So there was a big parallel between where I grew up in Salt Lake. But then around 17, I moved to Utah to go to school. And I always get the question of why Utah, because I'm not, I didn't have any affiliation with the state or anyone there. Nor was I involved in the church or anything like that. So it was actually just a little bit about the landscapes, the place itself, but also because the program that I was interested in was ranked among top 30 in the country, and it just seemed like it wasn't going to be that really cutthroat, intense environment like maybe some other schools in the U.S. It seemed like the right place. Um, and honestly, I toured a year before I ended up at Utah. I actually toured many schools in the U.S. While I was still in high school in Bolivia. And I remember that the only place where I really felt welcome was Utah. Like the recruiting team was phenomenal. The international office really actually took their time to give me a tour and say, Diego, this is what you're going to be able to do with us. Um, like we have big research programs, and if you come to our school, you're going to find a lab pretty easily. And then I toured Stanford and Berkeley, and like you were just another guy,
Yeah.
you were just one more person that was dreaming about going into the programs. And so I think that was a big thing for me. But in terms of what I grew up doing, I actually grew up skateboarding. As you said, I was always with my skateboard, and that's because I think I learned how to skate before I learned how to ride a bicycle or pretty much anything else. Which is really unusual because there isn't much skateboarding in Bolivia. I mean, it's just not. Our streets are cobblestones or dirt roads, so we don't have the skate parks, and so for me, that was really cool to see in the U.S. Obviously, like the U campus, right? It's a skate park in and of itself. There's just so many cool
Yeah.
places to skate.
Yeah. It's definitely it helps that we're like on a mountain, so you
Yeah,
definitely from the dorms you could definitely get to class like pretty fast on a skateboard. I'm surprised that it wasn't pretty popular in Bolivia because you said you grew up in a colder environment. Did you do any kind of like snowboarding, or was that available? Did it snow there?
yeah, that's a good question. I mean, paradoxically, there isn't. So we have snow. We can see snow on the mountains. So we live in the mountains, but we see taller mountains, which are the Andes. Which are 6,000 meters above sea level, so over 15, 16,000 feet, and you can see
Okay.
snow there. But it's not our culture to snowboard or to ski. It's not really a thing that people do in Bolivia. In Argentina, yes. In parts of Chile, yes, but not in Bolivia. But funny enough, my dad, who had lived abroad and traveled abroad, he actually tried to ski with my brother and I, and we actually would drive up to the mountains with his old skis that were like, I don't know, like super long and pretty
Yeah.
primitive skis, and actually it's more like ice. There's not even snow. It's like a
Oh
glacier up
wow.
there, and the slope is like this, like
Oh yeah, I'm cool. I'm cool. There's definitely been days at the U where like the rain would like freeze over, and if you just stood there, you would just slide down campus. So yeah, I'll pass on that.
Same.
You grew up in Bolivia. How close were you to the Amazon jungle?
Not that far. It's maybe so it's tough to define the jungle because if you drive down the mountains like maybe 3 hours from the city, you'll already be in the jungle. But the Amazon forest, the real Amazon forest is pretty far. Like you have to take a
Okay.
plane, and the difference I think is the ecosystem, so the altitude, the fauna, so the type of animals that you'll find. But Bolivia does have 2/3 of the country is all forest. So, and arguably one of the best, maybe one of the wildest. And I don't know if you've seen that movie with Daniel Radcliffe where he gets lost in the jungle, and that's all in Bolivia. It's
Oh
based on a true
wow.
story. Yeah.
The reason I asked you that question is because I actually know somebody from Bolivia as well, and
Really?
I think yeah. I think you may not remember him. He worked in like the little like I want to say like the little cafe section at the U
HC, right?
in the 8C. I forgot. Yeah, he worked at like the little store that they had right there. And
Yes, I
he
do. I do.
yeah, exactly. He used to tell me stories about like because he lived close to the jungle, and he would tell me stories about like I guess it had like he said the way that I remember him telling me a story about the parrots, and he said the way they look at like the zoo is nothing how they are in the wild. He said they're way bigger and they're loud, and he said if you like go into like the forest, a certain section, you can like hear them screaming. But he said it's like the way he said it kind of like scared me a little bit. He said it's nothing to really be. You're not supposed to go in there by yourself, basically.
I, I, I think I would be more scared of going into the jungle because of insects and diseases
Yeah,
than like a leopard or like a, like
yeah,
I think
something like
those little guys are actually the worst compared to like bigger animals that are also
yeah,
scary,
beetles.
but maybe
The beetles. He told me about the beetles that are out there. So he spoke
I
about how you got it, how you came to the University of Utah, but afterwards, like I said, you went to Princeton and
yeah.
Harvard. How did that all happen?
Maybe just to give you more context, the reason why I studied science wasn't really because I loved science per se. In the U.S. you need to go through a science program before you go to medical school. Not necessarily like you could do music or film, but then you also have to take those pre-medical courses, like in biology or in chemistry and physics and math before you go to medical school. And it's actually really helpful because you also need to take the MCATs to get into the program, like medical program. But
Hmm.
and one of the requirements to go to medical school in the United States is to actually do research. You need to be in a lab and do experiments and ideally publish an article, at least be an author in an article. But as I went into it, I started to really enjoy that. I liked the idea of working in the lab and coming up, with creative ways to solve really complex problems that no one else had solved. Even as an undergrad, I remember finding that pretty cool. And the U, as I said earlier, like had so many different labs that actually was really amazing to be able to find something that was interesting. And so I was working in two labs simultaneously. So after, between courses and after class, I would go to one lab, and then every other day with my skateboard.
Yeah, yeah.
um, and so that kind of propelled me into let's not do med school, let's do science. And the more I got into neuroscience and understanding how maybe the brain works, which we'll talk more about, I thought I like working with mice. There's a lot to do with them, but what if I worked with a higher model, like a higher level model, like monkeys, like non-human primates? And at that time, I started to screen. This is during my junior or senior year. I started to screen all the labs in the U.S. that were working on non-human primates doing neuroscience, but that were maybe in the areas that I would be interested in working, which were at the time a combination of like hardcore neuroscience and a little bit of psychology and cognitive biology. Um, and I found this lab at Princeton, which was amazing at that. And they had a really good publication record. And so I actually just sent the guy an email, the PI of the lab, and I said,
Wow.
you know, I'm doing this at Utah, and I actually, throughout my time at Utah, I traveled a lot to present my research in different conferences, and I got to publish two articles. One of them as the first author, which is highly unusual for an undergrad in terms of productivity. Also explains why my grades weren't the best, but I was productive in the science end. And this guy replied and said, wow, I'm actually incidentally looking for someone with your profile. And it was just amazing. I mean, that was really awesome. So as soon as I graduated, I think I spent a couple of weeks moving out of my dorm room because I was a residential advisor, so I had to wait for everybody to leave.
Okay.
And then I moved to Princeton, and that was a change. I mean, you know how it's like when you leave college and then you have to adapt to a life where you don't have the cafeteria to make friends, you don't have the dorms, and that was tough. I mean, the science was cool, but the life was weird after Utah, to be honest.
Oh wow.
Um,
Do you remember the last time we saw each other at Utah?
I do, I do, I do, I do, I do. Actually, the last time I saw you was when I was visiting from New Jersey, from Princeton. I came for a
Okay.
wedding after a couple months after I had left Utah, and we ran into each other in Research Park.
Yes, we did.
You
Do you
remember
remember
that?
do you remember that huge tarantula that like it was like
Yes.
it was a huge tarantula. I was like that came out of nowhere.
I remember we were talking, and I saw this. Yeah, and then you pointed at this knot, like it was this big.
It was huge,
Yeah,
huge, huge. I've never seen one that big since that day. Nobody believed me that there was a tarantula
I
like
was
that
there.
on the campus. Yeah.
That was really that was so unexpected. It was so cool to see you there. I mean, I wish in this podcast we could, like maybe I'd love to hear. I don't know if now or at some point, but I mean, your stories must have been so cool going, from the football team to what you did afterwards. And now I see what you're programming, and I mean, we're gonna be exchanging more as we get into the science of it. But
Yeah.
I mean, your background is also super fascinating.
Yeah, man, I planned it. Believe it or not, I had a clear idea of like what I wanted to do since I was about 12 years old. You
Mm,
know, um, I knew that football sports wasn't the end goal for me. I always wanted to pursue. I don't know if you saw that I played the guitar as well. I played classical guitar at the University of Utah. Did you?
no, no, I had no idea.
So I play guitar, and if you look into it, a lot of software engineers are they play instruments as well. They're musicians, so they play. You meet a lot of
Ah,
guitarists, a lot of pianists. So yeah, I went to the software engineering field. It just is what my passion was, and I told
mm,
myself when I was 12. I said, when I'm 30, I'm going to start my own business. So I founded a club at the University of Utah, the
yeah.
boxing club, and I only founded that just because I wanted to get some experience managing people. I wanted to learn how to invest my money into a project and see
Mm,
it grow. And it definitely it had a big impact on me, man, because I saw that it wasn't all about the sport. Like a lot of those people, similar to what you said your experience was at Princeton, a lot of people at the University of Utah, they went years without having like those strong relationships. They didn't have
mm,
relationships, romantic relationships, and a lot of
yeah.
people met their spouse on the boxing club. A lot of people met their whole social
Wow.
in the boxing club. So that kind of
That's
and
amazing.
and I had there was like people I would meet that they would recognize me, but I wouldn't recognize them, and they would see me around Salt Lake City, be like, "Hey, man, thank you for founding that club." I knew
Wow.
for that. So
That's
it's
remarkable.
yeah, yeah, little things like that that you don't think about. It makes a big role. It plays a big part in people's life.
For
But
sure. For sure.
yeah, man, go ahead.
And in that respect, do you feel like you maybe. Because, I mean, through you, I remember I met a few other players of the team, and I remember that. I mean, they were just playing football, but I mean, you seem to be a rare species in that bunch a little bit now because I don't know what those guys ended up doing, but you're doing engineering, the boxing club, I mean, leadership, and now the podcast. Is it a trend that people do this, or you find yourself to be one of a kind
I'm
there?
also yeah, I'm competing in boxing. I do amateur karate. I do a lot of different things, man. I think that put it like this, what I've learned. You've probably seen the same thing, dude. I mean, what I've learned is that it's similar to people that are in the military. A lot of people who do start businesses or people who do, I mean, they excel in other areas of life. They usually have like a military or athletic background.
Mm,
But just in general, though, man, I mean, most people that you're around, they just don't have a higher vision. Some people might be playing sports because their parents put them into it. Some people might be going to
yeah,
school because their parents put them into it. And then just, look at what you're doing. Most people aren't really like this isn't it. I'm sure you have a project that you're working on right
yeah,
now. And then after this project is done, you're going to have a whole nother project.
for
That's
sure,
who you are. And that's what's going to keep happening for your entire life. And I kind
for
of
sure.
been in the same mode as you, man. So yeah.
When you're driven and you have ambitions, then nothing that can stop you. So I understand that. I understand that, and that's also going back to the discussion that we were having about going from one place to another. I mean, when I was at Princeton working with these monkeys, and that's a story in and of itself because, working with monkeys wasn't so easy as I thought. I mean, these guys, funny enough, I was actually called the Monkey Whisperer. That was my nickname at Princeton.
That's
Because,
a sick nickname.
because the monkeys, we work with two different kinds of monkeys. The old African monkeys, which are mostly rhesus macaques, which are pretty big. I can't fit in the camera, but they're large.
Wow.
And also the New World monkeys, which are predominantly from South America, which grew up to like this big, and when they're babies, they're about this big. They're like little tiny guys. And the difference between both of them is that they've evolved to communicate in different ways. And by communicate, I mean literally exchanging turn taking is what it's called. It's when you speak, I'm listening, and when I speak, you're listening. So this ability to turn take is something that evolved in the New World monkeys in South America because they make vocal sounds and the other ones listen. And we, at the time, didn't really understand what parts of the brain were actually being activated during this neuronal activities. And so this is sort of what we were trying to understand by listening to the vocal sounds, learning what those vocal sounds were, understanding what different kinds of calls they made from a vocal point of view, and at the same time recording the electrical activity using electrodes that had been implanted through surgeries into these animals. But going back to the point of why I was called a monkey whisperer is because when you need to grab the monkey for an experiment, the monkeys live with other monkeys, and you need to get them to come to the experiment, but they don't want to. So you have to use different type of rewards, and so I became really good at using the food chain as a resource. I would hold on to grapes if I really needed a monkey, and if the monkey really didn't want to participate in the experiment, I would pull off marshmallow fluffs. And so actually my pockets in my lab coat were full of treats. But that was tough. And actually it was an interesting time because nowadays, and because monkey research in the U.S. is regulated by the USDA, not the case, by the way, for other animal models, they became really pretty tight. Like the regulatory agencies became pretty tight on like letting labs do monkey work for ethical reasons mostly. And so actually more and more labs starting to like send their monkeys out to sanctuaries and getting rid of their research in monkeys. And so actually my old lab continues to do that, but I noticed that it wasn't going to be the right place for me to do a PhD or a postdoc or to dedicate my science. And so that was actually one of the elements that pushed me to migrate over to other research, which is why about a year and a half I left and I took a job in a lab at Harvard, where I was sort of doing my master's, but also like working in the lab. And the goal there was, and this is actually one of the most interesting projects I've done, which is to work on the rejuvenation of the brain. And so what does that mean? So basically when we get old, the brain deteriorates. This is called senescence in neuroscience. And this deterioration is basically the loss of cells. Like people who have Alzheimer's disease and other neurodegenerative diseases, they actually see a lot of cell death. And why this happens is still not fully understood. But what's known is that when you get old, you have less and less blood vessels in the brain. Because blood vessels are like branches, like trees. And so if you don't have as many branches, or not only the number of branches, but also the length and like maybe how arbored they are, this architecture has an impact because you provide cells with oxygen, with enzymes, proteins, with nutrients that cells need to survive. And so it's kind of like a chicken and the egg kind of thing. Like nobody really understands whether it's the lack of blood vessels and oxygen and nutrients that go into cells that create senescence, or is it that senescence creates the deterioration of blood vessels? But regardless, the point is that when you age, this is what happens. Your cells start to die, your blood vessels start to lose their architecture. And so we found a way in an experiment that I'm about to describe to you, which sounds a little bit ominous, you'll hear. But we found a way to kind of revert this process of senescence. So we were doing these experiments where we were literally stitching mice. One old mouse, imagine a person that's 80 years old, well, analogous to that, a mouse that's very old, and a very young mouse, or like a teenager in terms of human years. And we would stitch them. So basically you would see two animals walking around together, sleeping together, because they were basically sharing their blood vasculature. For 5 weeks. And during these 5 weeks, the blood from the young mouse would go into the blood of the old mouse and vice versa. After the 5 weeks, we would remove their brain tissues and look at them. So we would cut them in small sections and put them under the microscope and see if anything had changed by comparing the old mouse with the young blood compared to just an average old mouse. And that's where we saw the biggest difference, that the entire blood vasculature architecture of the old mouse that had been exposed to the young blood completely rejuvenated, in the sense that it already had a lot of branches, a lot more blood vasculature than the regular old mouse. So something was in the blood of the young mouse that made this old mouse young again from an anatomical point of view. And it wasn't only in the blood vasculature, but it was also in the number of cells in a specific type of stem cell that you need to be able to replenish your cells when they die. So now there were more stem cells that could potentially replace the cells that had died. So again, we don't know what came first, whether the stem cells came first or the blood architecture came back first, but they both had a positive impact on the mouse brain. We also saw that there was more activity of cells, like there was actually more cells firing in the mouse that had been exposed to the young blood. And so this is what we call rejuvenation. But you cannot, if people are getting old, or people have Alzheimer's disease or some kind of age-related disease, you cannot just go stitching them to a teenager, right? It doesn't sound like, from a therapeutic point of view, it's not something that you would be able to do in real life with humans. And that's when we thought, well, there's something in the blood. There's got to be a molecule of some sort in the blood of the young mouse that must have at least, 50 or 60 or 70% must be the underlying factor that makes this rejuvenation happen. And this was a very expensive experiment. It wasn't done by us, but somebody else does what's called a proteomic analysis, where you understand which proteins are being expressed in the blood, and compare those to the blood of an old mouse, and that's where you see very specific factors that are highly expressed. And the one that came out to be the most important one is called GDF11, and which stands for growth differentiating factor 11, but the name is irrelevant. The point is that this factor seemed to be the one that was making the old mouse younger. And so then the question was, okay, so then what happens if you just inject this molecule, if you can buy it, and then put it into the old mouse? Do you see the same effect? Mm-hmm. And so, lo and behold, that's what happened. We injected the mouse with this drug for every day for several weeks, and we were able to see almost, not fully, but almost the same impact as the blood. So now we had identified something that could potentially become a drug for a therapeutic purpose. And at that point, we knew that this was happening in some parts of the brain, but we had to do it in all of the brain. We had to really see if this had a positive impact in other areas of the brain, not just only the areas we were focusing on, which were primarily in the hippocampus, which has to do with memory and learning. And so, because when you do these studies in the brain, you have to cut the brain of the mouse. You have to use these machines called vibratomes, which is going to section. It's basically like when you cut meat at a butcher. You just basically slice it in little pieces, and you then put every slice on a glass slide, and then you put the glass slide under the microscope, and you take an image, which takes forever. So
how small pieces do you cut it into?
It depends. Yeah.
Okay.
So usually they go into 30, 40 microns in thickness. 40 microns, so it's 1,000th of a millimeter. So pretty small. And so in one adult brain, you can get hundreds of sections. And you don't only do it with one mouse. You have to do it at least with 10, 12 mice. And so you actually end up having to cut for days and days.
You can see variation in each piece. That much variation in each piece. I'm wondering, like, you're cutting it into these small pieces, so I'm guessing that you see like there's a difference between each slice, right? Like there's some kind of variation.
Yeah, yeah. The reason why that's so important is because maybe not from one slice to the next you won't see a big difference, but after 10 you're no longer in the hippocampus, you're in the amygdala, or you're in the cerebellum. You're in a completely different area of the brain, and so you really have to spend a lot of time making these cuts, and then you have to. I don't want to go into too much detail, but then you have to put molecules into them that fluoresce so that the microscope can see them, because the brain isn't endogenously fluorescent. So you have to put something that tells you what's happening in that tissue. And you have to actually wash every section with these buffers and these reagents, these compounds, for days, and then it's a very tedious and manual process. And some guy at Stanford developed this tool called Clarity, which basically makes the brain transparent. Translucent, literally, you can actually hold it and it's transparent. Oh, wow. And the purpose of this, so the reason why it's so cool, is, besides the fact that it's transparent, is that you don't need to cut it anymore. You can just make it translucent, add your antibodies and whatever fluorescent molecules you need to see it under the microscope, but you just don't waste your time cutting it and putting each section of the brain into a cover slip in a glass slide and all that. Mm-hmm. And so my lab at Harvard was like, you're going to go to Stanford and you're going to learn this, man. And you're going to come back and you're going to do it here, because we don't have the time to do this study. So they flew me out to the lab of this huge guy. It was amazing. I've actually had to work. I've gotten to work with the director of the Stem Cell Institute at Harvard, and now the director of the bioengineering program at Stanford, and like all these amazing people. And I was there for a couple, 2 to 3 weeks, and they taught me from A to Z how to do this. I came back to Boston, and I started doing that. And I started doing these incredibly huge reconstructions of the brain to see if this blood vasculature rejuvenation actually took place everywhere. And that's when my abilities came into a roadblock. And this is where it gets, this is where science is getting to. And this is why people with your background and with your interest at this moment are so desired in science. Because we have so much data, but we have no idea how to analyze it, how to visualize it, how to process it, right? I mean, biologists have been carrying around their data with their Excel sheets and their PowerPoint slides with thumb drives. And now we're sitting on 30 terabytes of data, and we're like, man, how do I even open a 1 terabyte image? I mean, I have no idea. I mean, you can no longer use a software. At this point, you either know how to break the data down either through some Python library, or you custom make it yourself, but it's like, we don't know at this point, right? I mean, and so we have these amazing images, but that's all we could do. I mean, we couldn't analyze it. It was too much for us. And our lab was not a computational lab. And so we kind of left it there. And this was actually the beginning of a company which now received funding. Well, now it's 7 years ago, but the company is a startup company in Boston that received several $1 million to be able to turn this into a therapy. So actually, I contributed. My experiments contributed to this, which was pretty amazing. But the person running the project, which was my immediate manager, left. She got offers from Johnson & Johnson, who started a company to be able to pay for these rejuvenation projects, which were being funded by Peter Thiel in California and other people, because those guys out in Silicon Valley are obsessed with these new revolutionary rejuvenating projects for I don't know what reasons, but yeah, so she left, and at that point I was like, I can either stay or I can leave. And so some people contacted me and was like, are you interested in doing a PhD? And there was this really interesting project happening in France at the Pasteur Institute. And that's sort of when the idea of going to Europe came about. I then took a trip to Hawaii to think about it, because it was like, fuck, man, do I really want to leave the US? I'm not sure. I was kind of like, I'd been there for 7 or more years, and the girlfriend, the friends, the everything, and
the girlfriend didn't come with you?
No.
Oh, okay. See, Europe's fun, man. I've been to Europe. I mean, it's a good experience.
You know, that's what my mom helped me a lot with this decision. She said, Diego, you know, like look at your resume. You have these great experiences. You went to one of the best engineering schools, chemistry. You know, Utah. You did Princeton. You did the monkey work you wanted to do. But like, I feel like your academic CV's here, but your life CV's here. And you know, I was never interested in traveling as much as I spent the summer teaching English in China while I was at Utah, but otherwise I had never really been around, and I wasn't super interested, you know, like culture museums and that kind of stuff wasn't really my thing, to be honest.
I'm the same
But
way.
say again.
I'm the same way. I promised a few people that we went to college with that I was visit them, and it took me about 10 years to go out to Europe and go out to the Middle East to actually visit them. I'm going to South Korea in July to actually hang out with
With Young,
exactly Young Lee. I'm
yeah,
gonna hang out with him and his kids. So
I
yeah,
can't
go ahead.
I can't believe I beat you. I thought you had already been there.
Oh, you've been there?
I have. I gave a talk at the U of U campus in Incheon, which is outside of Seoul.
Did you see Young when you were there?
I
Were
saw
you?
Young and I saw James Park, who you probably also remember.
Oh wow, man! I haven't seen James in a minute, dude. Oh, I forgot
Yeah,
about James.
yeah, he's
God,
great. He
how
has
long
a
ago was it?
this was before COVID. This must have been now 3 years ago exactly. Exactly 3 years
Wow,
ago. Yeah.
wow.
Um, and yeah, actually, because I had something to do in Japan, and James heard about it, and he said, "You gotta come out. You know, Japan is so close." And so the U of U campus there, they flew me out from Kyoto to Seoul, and then James gave me a tour, and then I saw Young, and I came to the school, and James has a really high position at the U of U Korea now.
Oh wow.
We met with the president of the university, and it was amazing. It was a great experience, and I think
Oh,
your story should also be shared. And so I feel like when you're out there, definitely would be
I
great.
should I should definitely contact James before I head
Yeah.
out. See what's going on. One of the things I wanted to touch base on was a question that I asked you was I hear that the brain is like it's always being compared to a computer. When I
Yeah.
initially uh, when I initially sent that question to you, I thought it out. I think I understand it. In software development, the reason that an application works is because it's a collection of services, and that services they are umbrellaed by like one overall system. So I'm
Hmm.
guessing that brain, like software engineering, it's a collection of services that work together for a specific function. You mentioned that people in my area, because of like I guess how the field is like evolving, there's more people who are software engineers getting into that area. Like what are they focused on specifically? Like what is their responsibility with this data, and what
Yeah.
role do they?
Totally. So there are two questions. So the first one is how is the brain? What are the parallels between the brain and a computer? And I think one that I believe in, and I think is the one that would make the most sense, is the fact that, and maybe this is old computing now, but you know this binary idea of when cells are, you know, the way in which the brain works is we have different types of cells, but the predominant one is called a neuron, and the neuron is basically going to communicate a signal from one cell to the next, and it's either activated or inactivated. So this is how we can think of it as binary. It's either on or off. Much like I guess many operations in a computer, at least in the old version of a computer, and so or program. So this is I think the closest one of the parallels. The other is that there's a network, and there's this a wiring of the brain in which the brain is structured or built. You know, because one cell is going to pass on something onto the next. So it's like a circuit, I suppose. And by looking at this circuit, you can actually see how the signal can go from one part of the brain to the other. Now, there is plasticity, which means that the brain is flexible. So this circuit is not always bound there for, as far as we understand, forever. It's quite flexible, but it's not that flexible where a neuron that's in this side of the brain will suddenly connect with one and the other side. I mean, the brain is wired already. It's pretty much set in stone. Not all the parts of the brain are set in stone when you're born, actually. There's one part of the brain which I studied heavily, extensively during my PhD, which is the cerebellum, which sits in the back of the head. And actually, when you're born, the cells in the cerebellum haven't even formed synapses yet because they haven't even arrived at their final positions. They're still moving around. It's a process called migration, cell migration. And that's why when you're born, you don't really know what's up and down or left and right. You don't know how to balance yourself because that part of the brain isn't functional yet. And as you're born during the first month or year of birth, actually, the cells are still sort of like moving to the right location in that part of the brain. And then eventually they grow their axons. And this axon is going to transmit this electrical potential, which is basically just ions that flood into the cell. Like sodium, potassium pumps, which are going to create a flow that's going to be sped up by a sheet, kind of like insulates the cell. It's another cell that's going to help the cell pass this electricity faster until it reaches the end of the cell, and this is how it's going to be activated, and then it passes on the action potential into the next cell through neurotransmitters. But yeah, this is, I guess, long story short, I think this is the way a computer is close to the brain. It's like it's a circuit, and there's a binary mode in which a cell is either active or inactive.
And I guess making the brain transparent allows you to see like that, see how those cells communicate, right?
Unfortunately, no, because when you make the brain transparent, you are fixing the brain. You are killing it. And to be able to see this potential, the brain needs to be functional. It needs to be alive. So
And how do you see that then?
Yeah. So there are a variety of ways. One is it's called a cranial window. So you remove a part of the skull of the animal. You put a little window, and you hold the animal, which is usually a mouse. You put the mouse against an objective of the microscope, which has a laser, and the laser is going to excite the photons, which are going to be actually proteins that fluoresce inside of the cell that is in the brain. It's basically you work with mice that have been engineered to be able to fluoresce every time the cell is active, and this is going to be captured by the detector, which is also the objective of the camera on the microscope. The problem is that the depth at which you can observe these events is very small because there is something called light refraction. I mean, first, the light cannot penetrate tissue. It can only go so far. Maybe 700 microns at the most, which is not even, depending on the size, the body of a cell is usually about 10 to 15 micrometers. But they grow really long protrusions, and so you cannot see more than a few, maybe 10 cells, deep maximum, and the brain is made up of trillions of cells probably, so millions. So basically, it's a challenging thing to observe these events happening in real life. And this is why the brain mapping field is divided into different segments. So people that do brain mapping at the macro level, so they know holistically what parts of the brain are active when somebody's talking or thinking about some images or looking at a movie that's sad or having different emotions. And they do this through just looking at, they use large machines that basically measure the blood flow going into different parts of the brain. This is functional magnetic resonance imaging. But they cannot actually see how the cells individually pass on electrical currents from one cell to the next. The resolution isn't high enough. And so this is why brain mapping at this macro level is very different from brain mapping at the micro or nanoscopic level, which is what I do, or did actually. The macro level allows you to see things live in living conditions. The micro level allows you to see things in living conditions, but very limited with this cranial windows. And the nano level, which is what I did, unfortunately doesn't give you the temporal dimension. You can only see things in 3D, not 4D. So you can see them in without moving. You can only see them at one point in time, unfortunately.
You mentioned about having an older mouse and injecting that older mouse with younger blood, and you basically can see that the that mouse was basically reverse aging on some
Yes.
level. I'm not sure if you remember. Have you ever heard of this game called Horizon Zero Dawn?
No, what is it?
Just this conversation, it's reminding me of this game, and
I don't know if that's a
it
good
talks
thing.
about like it's talking about in the game it's Earth and it's Earth 1,000 years after like civilization falls apart, and all animals, most like 80%, 90% of life forms have been replaced by robots, and these robots look like everyday regular animals. And there's a system on the planet, a computer system, a collection of different computer systems on the planet that are they operate alone. They don't need a human to basically help them operate. But what they do is they keep the planet alive. These computers, they keep the planet alive, and they make sure that like basically life doesn't disappear
Wow.
100% completely. And I want to say after 1,000 years, the people who are scientists, who were engineers, they return back to planet Earth, and it's the same people who left. So they actually are like over 1,000 years old, and
Cool.
they found a way to basically make their cells prevent their cells from aging.
Ah,
So it's just weird having this conversation with you, and then also having that reference because it's kind of like we had that we're connecting the brain to a computer, and on this game, the computers are essentially controlling planet Earth, keeping it alive, and then you have these people who are immortal in their brains and whatnot. Do you think that's the direction that science is ultimately trying to move to? They're trying to prolong life, um, so to speak.
I believe that some people are interested in that specifically, prolonging life, making life longer. In some cases, even becoming immortal. I know that, and I've heard or read extracts from billionaires who are investing in this. They are investing their own money into research that
Yeah.
can go in this direction. However, I think from a scientific point of view, at least us in the scientific community, we care more about either the quality of life, so not that you live longer, but that you live better while you're alive, or to use it in therapy for people with Alzheimer's disease. For example, when you have Alzheimer's and your cells are dying, if there are many ways, by the way, to tackle Alzheimer's, and unfortunately there isn't one yet that works really well, but one potential approach is to replace the cells that are dying. And so how do you replace cells that are dying? You could, in theory, inject yourself with cells, but the brain might reject them. So you just make your own brain create more cells, but the stem cells that the brain uses to create more cells are gone by the time you get Alzheimer's. And so by injecting this, in principle, patients with Alzheimer's with this drug, they themselves are, with the blood vasculature, it's going to induce more stem cells, and these stem cells are going to, in principle, become the cells that are, in principle, dying because of the disease. And so I think for us, at least at Harvard, that was our goal. It was to be able to circumvent the damage that's being caused by this neurodegenerative disease, rather than let's just make people live longer, because actually that's not what our research shows anyway. We never actually understood whether the animal that has this treatment lives for twice the age. We have no idea. I mean, we never looked at that. We just wanted to see whether this change was significant from an anatomical point of view. And later, other people will have to test whether it's actually long lasting, because maybe we inject you, your brain becomes better, but then you go back to where your old state within a few weeks. So does that mean that you have to inject yourself every day for the rest of your life? I mean, fun fact, actually not so funny, but some people have found this drug in the market, in the black market, and now there's a YouTube community of people injecting themselves with this thing called GDF11, and they're like, I'm patient 1 or I'm patient 0. And this is, my old
GDF11.
GDF11.
Okay.
If you're gonna buy some. GDF11 for sale, community aware forum.
I'm not gonna get into that. I'm gonna look it up like later. I'm not gonna,
I hope so.
backtrack and try to inject myself with some drug that hasn't even been, verified. I
Yeah.
was gonna ask you that though. I was gonna ask, like, when do you think well, you answered that question. I was gonna say, when do you think humans are actually going to start using this, or when is it going to be? I want to say like certified. It goes through all those channels where it's actually made
Yeah.
publicly accessible. We know that it works on mice. I'm sure there's probably been some tests on monkeys as well. So it works on some level. When is it
Yeah.
going to be
Sure.
available to people?
Um.
And what's blocking it? And what's
Yeah.
preventing it from getting out there now?
Well, I think there are a variety of forces and drivers that are going to push it, and forces that are going to prevent it from getting anywhere at this point. One of them is that the research from an academic, from a basic fundamental research point of view, it's still unclear whether there are no side effects on the mouse, because growth factors are in young animals, in young people, because they need them, because cells are dividing. That's actually the real biological purpose of these molecules, is for cells to continue to divide and to grow, for you to grow as the human that you are. That's why you have them when you're a kid and not when you're an adult. When you're an adult and you induce growth factors, you could potentially cause cell division that can lead to a tumor. And this is because cancer is basically the result of uncontrolled cell division. This is what happens. This is why you get a tumor, is because cells in that part of the body continue to divide uncontrollably, and this is why you have a bump or a tumor. And so these growth factors have been described in the literature to be inducers of cell division and therefore tumor growth. And so until that can be understood from a basic point of view in mice, I think it'll be very difficult to push this even to clinical trials. And even clinical trials, with the exception of what we've all heard about the COVID vaccines, it's a process that takes roughly 10 to 15 years. And so I think it's realistic to, if we ever think of a GDF11 as a potential therapeutic agent, it's going to take at least 10 to 15 years to ever hear of it as a potential drug. Then you have insurance companies who may want to cover it or not for some diseases, and at that point it will depend on the indications that it's been tested for. Most likely just Alzheimer's. So it would be for like patients with neurodegenerative diseases rather than for people to, use it themselves.
Okay, that's a lot sooner than I was expecting. Could you explain what, I think we're kind of talking about it a little bit, but could you explain what connectomics is?
Sure,
Connectomics?
sure. So that's actually something that I did mostly when I was in France at that point.
Okay.
But funny enough, at Harvard, because the microscopes that we needed to do this were there, and so actually I was coming back and forth between Paris and Cambridge, nonetheless.
So you're not in France anymore right now?
I'm still in France, but the work I did with connectomics was actually done half at Harvard, and so actually I was always flying back and forth in the end. So actually the decision to come from France was sort of a half and half. But so connectomics, so omics, if you've heard of omics, we've heard about genomics, we've heard about transcriptomics. These are just ways in which we can fully map something. So genomics is mapping genes. Transcriptomics is mapping of the transcription, which is when the DNA becomes RNA and then a protein. Okay. Connectomics refers to the mapping of connections in the brain, which basically means coming up with Google Maps of the brain. So basically, you could be in a cell and you actually can see what's in front of you, and you can, basically move from one cell to the next, and then look to your left, to your right, behind you, above and below you. You can see all the cells around you. And connectomics requires an imaging method, a microscope method that's extremely powerful, so that you can actually see what's happening inside of every cell. And only when you have looked at every cell and all the cells that are around it can you actually build a connectome. The problem is that, and this going back to the whole why we need computer scientists like yourself, is because we come up with a lot of data. I mean, I had dozens of terabytes of data that we struggle to handle because we're not computer scientists, and we use pretty primitive ways to analyze data from our side. And actually, maybe I'll describe the method a bit more. So basically, to be able to look at these cells at this resolution and look at them in 3D, you need to cut the brain very thin, as I was describing earlier, but much thinner now. Now it's 30 nanometers instead of 30 microns. So now it's like a tiny little sheet of paper. I don't know if you ever did this, but did you ever, like when you were a kid, draw little things on the bottom right or left of a book, and then the next page you draw the same but like just slightly different, and then at the end you could like shuffle through the pages and you could see kind of like a storyline, right?
Oh yeah, I definitely did that.
Yeah, I think
definitely.
we all did. So that's sort of the concept. So like we basically cut the brain until in one image you don't see you see an image in one page, you turn the page, it looks exactly the same, and then exactly the same, but in the end if you shuffle all of them, you can actually see the whole thing looking different. This is exactly what we did. We cut the brain so thin that if you just turn the pages, it looked exactly the same. But if you look through the entire block of images, that's when you actually see all the cells that were everywhere. But they come in black and white because the brain is not in colors. And so I actually hired an army of 25 students. And by hire, I mean, got them really nice recommendation letters and told them that they would get into good grad schools if they work with me. And they colored every cell manually, one page to the next. Imagine like doing that for your notebook drawings. Just like you color it, then the next page you color it. It's through thousands of pages. I mean, this took years. And now we have this color map. Everything's in colors. It's like red, green, blue, pink, blue, every color. And every cell has a different color. And now we can actually, and this is actually a parenthesis, this is also where we need computational help. We use AI algorithms, and Google has actually been super helpful at this. Help us design codes that can help us color the cells automatically so the students don't have to do it anymore. So you basically show the computer, oh, this is what a cell looks like. These are the boundaries of the cell. And through machine learning, the machine is able to basically understand that that there's a filling, there's a certain pattern in the filling of the cell, colored. We call it segmentation. But and now computers can actually do it, but they do have a big margin of error that needs to be manually corrected, and so it takes time in the end. But
What is that? I mean, I get the AI portion of it, how AI works and how data works on that side, and basically seeing the relationship between that information. But what is being left out? What's being read incorrectly on
yeah,
on that side?
I love these questions. I love these questions because I love the technical side of it. So the images of the brain are phenomenal. I mean, the human eye can actually tell the boundary of the cell, and so the beginning of a cell and the end of a cell. Even if there's a little gap, a pixel or 2 pixels that are missing, like black or white, saturated, oversaturated, but the computer doesn't see that. The computer assumes that that's actually the opening. This is where 2 cells actually connect. It's like a tunnel. And so the cell will color the 2 cells with the same color because it assumes that that's a bridge between the 2 cells. It doesn't put the context. And so these are called, there's a name for this error, but it basically is it's like it's a watershed error of some sort where the segmentation goes into the other cell.
The name of the error is what? Because I get errors all the time while I'm working.
On the code itself, on the coding itself, I don't know the actual error, but it is it has to. There are 2 types of errors. Either when the computer thinks that the end exists, but actually it doesn't. Like it's just the growth. It's just a pixel that looks too dark, so the computer thinks the cell just ended. And I'd be happy to share more about this with you if you ever have any ideas, but. That's one of the errors, or when there's a pixel that's open, or the coloring of the staining of the cells to make the images actually didn't work perfectly, and so there are a variety of those 2 errors are primarily the errors. And so once we have colored those cells, we export them as 3-dimensional objects, and we can open them with Blender or 3D Studio Max or Maya from Autodesk. And we can rotate around the cells, and we can make videos to basically visualize what's happening in the cells in the 3-dimensional view. Again, we don't have the temporal dimension, unfortunately, because the brain is dead. We've fixed it. But we can already learn a lot because we've never actually seen what a map of the brain at this resolution, where like you can actually see how a cell interacts with another cell. And actually,
Has there ever been any studying on a live brain? Like, I'm sure somebody has tried to do that. Has there been any? And
yeah,
what have been those results?
there have been plenty. Never at this resolution. You'll never, unfortunately, be able to reach this resolution with a live brain because the microscopes that we use are called electron microscopes, which use electrons. You bombard the tissue with electrons, and you need to cut the brain to be able to see at that resolution. So to keep it alive, you cannot cut it. You cannot kill the brain. You have to keep it alive. And in this case, you are limited to using a different microscope method, which is called light microscopy, which uses photons instead of electrons. And so we can look at the brain and the circuitry of like C. elegans, which are worms, or flies, or fish. Zebrafish when they're small, when they're babies. You can actually see the entire spinal cord firing with neurons, and you can see the colors, and with an impressive resolution, even in 4 dimensions. Well, 4 because you have the time, but 3D because you have multiple detectors on the microscope. So you can take an image from this side, and you can reconstruct the entire fish as it's basically developing. It's amazing. It's incredible. And you also end up with terabytes of data. But what's come out of it? It depends on the question. A lot of people have modified the fish to not have a protein X. So you genetically engineer the animal, in this case a fish, so that it doesn't have a protein X. And then you see if those cells that need this protein to survive are able to transmit a signal. You can modify any gene you want, actually, at this point. And you can see what impact that has in the function or the structure of the animal in living conditions. And you have to do it dozens of times and actually see if your results are statistically significant. But as you can see, at every step, in every part of biology right now, we need computer scientists. And this is why if you go looking for jobs right now in science, half of those jobs are going to be like, we need somebody that understands how to write code, either in Python, MATLAB's in the past, even C++, but just generally understanding how to use novel, automated, like machine learning based methods for segmentation or image analysis. I think is huge.
Okay, I definitely should check that out. And I know why they're using Python. Python is usually, we use Python on our APIs, and that's because Python it helps you basically, it helps you with data, essentially.
Yeah.
Well, I was going to say, you did some research with Jeff Lynchman, correct? And this is
Yeah.
his Connect Thomas' area of focus.
Exactly.
I kind of get, well, I get exactly like what connectomics, what it means, but what is ultimately the goal of it? Like, say we have a very good understanding of it. What is ultimately the goal of this field? Because in his video that you sent me, it talks about rewiring the human brain. Like, what is the goal?
That's a good question. It's a good question because it's a difficult question to answer, and it's difficult because many people see different goals in it. And in our case, in our lab in France, connectomics was more of means to an end. It was to actually identify a structure that we can talk about later. It's called a TNT, which is a new organelle that nobody sort of had seen before. But for Jeff and the people that we consider the founding fathers of this field of neuroscience, because it's fairly new, it basically means understanding how every cell connects to the other so that we can actually design a diagram of the brain. And so in the goal of that is actually to, first of all, I guess appreciate a little bit of how the brain is constructed, because you have very few people in neuroscience that are only devoted to the idea of seeing how cells actually are connected to one another. And Jeff is, one of the greatest scientists that I got to work with, and I got to see how he has this incredible interest in the original. You could call him the founder of neuroscience. It was a Spanish scientist named Santiago Ramón y Cajal. And this guy actually used this little microscope, and he would have these slices of the brain under his microscope, and he would actually use a pencil and a piece of paper and draw out neurons. So he's the one, Ramon y Cajal is the one who discovered the neuron. And this is 100 years ago, you know. He wrote this in 1888, if I'm not mistaken. He got a Nobel Prize of physiology and medicine at some point. And I think Jeff is kind of like using the technologies that we have available nowadays in connectomics, in electron microscopy, and machine learning and artificial intelligence to be able to create a map of the brain with the purpose of understanding how these brains actually connect to one another. Because maybe disease, so normal functions and diseases in principle should have their footprints should be in that diagram, basically. So if there is an issue with maybe a patient that has some kind of disorder, in principle, is there a difference between that and a normal person? But yeah, again, it depends on how you see it, because some people are doing connectomics on a very specific region of the brain because they're interested in actually seeing how those specific cells communicate with others. So in our case, we were interested in the cerebellum, which I was talking about, is involved in balance and posture. So it really depends on the lab. I think connectomics really addresses different questions depending on the lab using it.
Turn your light on, Diego. I can see you, but it's starting to get dark over there. I can tell.
Good. Too bright now?
At least I'm not the only one that's like broken. I'm looking at myself. I'm like, man, I really need to get this light situation figured out.
How are you not sweating right now?
I'm trying not. I think I am sweating. I kind of did this a few times. Wipe my cheeks.
How was that? Parentheses. How was that playing in the incredible? You must have played in some really cool stadiums, right? I mean, when you played football back in the day.
Yeah.
I mean, those lights. I mean, you must have been used to the pressure. I mean,
it's funny that you said that, because as you were talking, a memory just popped into my head of seeing those lights and being on the field or whatnot. Honestly, it just adjusts, man. When you're in a situation, I was watching a podcast and the guy was talking about how to prevent aging, and one of the things that he said was, like, I guess combat sports does it. He said because when you have, like, your brain has to calculate, like, the speed of a punch or the angle of a punch, and when you do that for a long period of time over years, your brain it slows down the aging process. And I think that to relate that to football, when you're playing football or any kind of, like, combat sport, when your body is in danger and you have, like, guys that are 300 pounds running faster than most human beings, and all these things are coming after you, you kind of, like, you zone in. It's like you have tunnel vision, so the lights and all that stuff doesn't really affect you.
Because you're a big guy, I mean, but there must be really enormous people playing football then, if you think of them as big guys.
They're ridiculous. There's guys, the one thing I noticed about playing ball is that you meet, like, a one-of-a-kind human being. You meet people who look like they shouldn't be as strong as they are. There's people who are, like, 170 pounds lifting, like, 400, 500 pounds,
Wow.
And then there's guys who are, like, 300 pounds who can do back flips and splits and run like the wind. So you get to see humans do incredible things. So that's the one thing I'm really, I'm glad that I saw it, because I know it's capable. And because I know it's capable, I can teach it to other people. I can say, hey, this is possible. You can do this if you work on it.
Interesting.
Definitely, because most people don't have that experience. They'll see guys, they'll see a regular human being who has, like, more musculature than the average person, and they'll think, like, oh, this person must be on steroids. But me being around those type of men for so long, most people aren't even aware of what they're physically capable of. It's just because we live in this system of the society we live in, we've kind of just adapted to this way of life. But if we weren't in this system and we were in those, like, high intense situations, like, your body, you would change.
Yeah.
Your thinking would, your body physically, you would look different,
For sure. Wow, that's fascinating. Another question was, I mean, the U is a university that's loves football, right? Like, they love the team. The Utes are amazing.
Yeah.
How was that for you in the school? Like, you must have gotten a lot of attention, I presume, right? Like, it's being a football player. I mean, how was that what's that experience to be a football player in a school where football is so important?
Well, you know how the colleges have like fraternities, sororities. The football team is, even amongst all the sports, the football team is like the number one fraternity. So when you do play, yeah, when you do play football, you're instantly a celebrity in Utah. Even I think you're even a bigger celebrity than like the BYU football players. You know, the Utah's football team is like one of the best football teams in the country.
Yeah.
And yeah, man. I mean, there was moments where I remember I was like, I would be on a date, and then there would be women who would come up to me while I'm on a date and try to talk to me while I'm sitting there at the table with my date. There were situations where I was like on public transportation. The bus driver would stop the bus and come back and shake my hand, and then walk back to his seat and continue on with the bus route.
Wow.
Yeah, and man, and the kids love you, man. Like
I bet.
we did a lot of things in the summertime, man, and then like yes, you get a lot of attention, and you get like a lot of haters as well. But I do remember the kids, like kids, they would genuinely like, it was like they saw Superman,
Mm,
you know,
for sure.
like they really, and I was so aware of it because I remember being that kid at some point in my life,
Mm.
and being there, taking those pictures with them, signing the autograph, having a small talk with them, like we're talking, like that really plays a big part in their life. Like they really appreciate it.
You're really in a
So
position where you can inspire them, people in general.
exactly. Yes, they're really open. Oh, exactly. They're so open and receptive to anything that I have to do or say. So that was the biggest. That's one of the biggest things I was aware of, that I was and that I appreciated, and I didn't
Mm.
want to try to. I didn't want to try to exploit it. You know, I
Hmm.
wanted to. I was really mindful of what I said when I had that many eyes on me. And I wasn't even one of the stars. So yeah, I mean that's probably the biggest thing that I've actually seen. I remember playing and being a part of the football team.
Wow. Yeah, what an what an incredible life experience. I mean, very few people get
Yeah,
to do that, right? Like, how many students, statistically, like percentage wise, like less than 0.1% of all the students
definitely,
that.
man. I tried to play professional. I had a workout. They have these like sectional combines. So I had one at like the Seattle Seahawks, that's a professional team, and then the San Diego Chargers, and I ended up not making it. But I remember telling people, I was like, man, if I wasn't playing football, I could have done study abroad. I could have been making money way, way earlier, doing what I'm doing way earlier. But at the end of the day, I would never take that experience back. Because you're right, I can never. There's nothing. I can never get that experience back. I can never.
Sure.
There's nothing in life that's like that. Most people never get to experience that. And I definitely. It was tough because you do have to wake up at like 6 a.m. at 5 for 6 a.m. practice, then you go to class and you have practice again, and then there are teachers give you a hard time sometimes if you're an athlete. I've had that. A
Right.
lot of like what you said at the beginning. A lot of teachers, they don't see, at least in well, before I get into that, a lot of teachers don't see a lot of athletes or football players doing what I'm doing, being a classical guitarist,
Yeah.
moving on to a software
For
engineering
sure.
role
For
and doing
sure.
things like that. And I've known a few other athletes that have done things similar to what I'm doing, and that went to medical field and excelled. But I forgot what I was leading to. I can't remember, man. But forgot, dude.
I'll
I
come
can't
back to you.
remember.
I'll come back to you.
God, I forgot. It was good, but anyways, yeah, man, it was a great experience, and it's not something that I don't miss it. I like having control and the freedom to do what I want in my life, but I do miss being around those type of men because when you play sports your whole life from like age 4 to 24, 25, it's like that's all you know. That's your identity, and then you get out into the real world, you realize how the everyday average human being, how they operate. And most people don't operate on like that level,
For sure.
physically, mentally, and focus,
For sure.
like your focus even have. So
For sure. For sure.
yeah,
For sure.
that part.
But speaking about rejuvenation, we've talked about this for a while. But one of the questions I always get, even though I'm not, besides the scientific side, I haven't really done much more research on this. But the question I always get is, what are the ways in which a person can rejuvenate without necessarily using this drug or putting, injecting yourself with young blood? And I didn't do this research, but I'm aware that there's a lot of really good research, and by good I mean in great journals and from great universities. I mean, good by social standards, let's say, because it doesn't necessarily, a study doesn't need to come from Harvard to be good. But there's a lot of research that shows that exercise in the long term actually has the same impact that maybe this drug can have in your rejuvenation of the brain. And so this is done with mice again. It's always done with mice, but nonetheless, because it's just easier to see the impact this has from an anatomical point of view in the brain later, because you can actually observe the brain afterwards. Calorie restriction is a huge one. So yeah, those two, exercise and calorie restriction, I think are the biggest factors of not of normal aging, but that you can actually change and eventually go through a natural, organic rejuvenation process. And I think for people like you that started, that did exercise continuously for a long time, it's like a normal part of your life,
It's who I am. If I don't do it, if I don't move, I get sick. I have to wake up at 3 and I have to go and do something physical way early in the morning, or else my entire day is it's off balance. One of the things that just came to my mind is a few years back when Instagram first came out. Not when it first came out, but a few years back, I found myself when I would get on like Instagram or I would look at any kind of social media, and it was negative. Let's say I woke up the first thing in my morning, I got on social media, and there was something negative. I realized throughout the day I would have more negative thoughts. So I was like, okay, now I started listening to more audiobooks, books that are around business or anything I'm trying to focus on. And I would think about those things unconsciously throughout my day. What I want to say, it would be cool if there was some kind of study to see how the brain operates, why the brain does that. Why does the brain take something, some kind of experience, and then it holds on to it for like, whether you're aware of it or not, it holds on to like that experience for like a number of hours throughout that day. You know what I'm trying to
Yeah, no, I see what you mean. I see what you mean. And I think that everybody has experienced this to a degree. I think this is obviously the interface between neuroscience and psychology. I think psychologists and cognitive neuroscientists have actually looked at this. But from my experience, I've just never actually found any study or done any research on this. But I definitely think it's important. I definitely agree with you.
It was just something cool. I was thinking.
Definitely.
One thing we mentioned on our last phone call, we were talking about how traumatic events kill brain cells. And we were more focused on like the physical traumatic events. Like obviously we see it in football with brain damage, and you had you see it with soccer as well, and boxing, because of the constant.
Sure.
But I informed you that a friend of mine was actually in an avalanche and witnessed someone die in front of her, somebody that she was in love with, and she had to go to therapy for that, and it was really traumatic for her.
Yeah.
So I'm wondering, do mental traumatic events, do they affect the aging process just as like the physical ones?
So actually, when you brought this up during our conversation, I did think a little bit about it, and I did do some research, and I found that even the research in this field is pretty, I would say, immature. It's still it's pretty young. Even at this, just to give you a sense of like how early on the research is at. There was this publication published in the journal Nature that said that these scientists from the NIH with Switzerland and Germany, they had found that in mice, when you stress the mouse with the shock, an electrical shock, you can actually traumatize the mouse, and you can actually trace what is the circuit pathway, so in the neural pathway that's actually being actually activated during this process. And it's one that's in part of the brain called the amygdala, which is somewhere in the brain, in the back of the temporal or frontal lobe. And what they saw is that the animals would respond in different ways. Some animals would actually find ways to avoid this electrical circuit. It's almost like they they inhibit it. So they're in the brain, in the cells there are two types of neurons. They're inhibitory neurons, and they're neurons that activate, which are called glutamate. So there are two molecules that actually play the balance of activation and inactivation in the brain. One is called glutamate, and one is called GABA. And so they would see that this stressful response would actually be inactivated by these inhibitory cells in some animals and not in others, which basically, for the first time, has been described. For the first time, scientists have actually identified what happens when people develop these kinds of stresses. But at this point, it's difficult to know how people, from the anatomical point of view, how you can actually stop that from happening, right? Besides psychological, maybe like methods, there isn't a way from an anatomical point of view to actually stop this traumatic event to maybe like evolve, overcome it.
It's just interesting because physically traumatic events, they happen within that instant, within the sport. It happens within that moment. But mentally tragic events can last you a decade.
Yeah.
And I've seen, people, their hair fall out, or white hair will turn white or gray. Obviously something's happening to the cells. The cells are dying on some level. So I was just wondering, just curious, like,
yeah,
what information's out there?
I don't know if the cells are necessarily dying. I just think that there are pathways that are being reinforced. Circuitries, like in terms of like neuronal circuits that are being activated, and they're not being balanced correctly. But
okay. Go ahead, my bad, man. I was. You about to finish?
No, I was gonna say, but I feel like the research itself is pretty early on. As you can see, this is actually one of the challenges that we face in neuroscience, is that there are just so many questions, and each question has, miles of depth. Which makes you pretty humble, actually, when you go into science. You realize that, actually, the more you try to learn about the brain, the more you realize you actually know less. Yeah, no, I think this is maybe what science is so challenging. It's just so much to learn, and it's too much,
Yeah, you really, with your last statement, really put me in like a trance, like deep thought. It's like the expanding universe. There's so much going on. That's just like, yeah, man. I wonder if we ever get to like the source of all this stuff.
Yeah, and actually, and I don't know if I told you about this, or if you saw this in the blurb of my resume that I sent you, but at some point I thought, I'm working towards this research. I'm helping people or scientists, the scientific community, understand how this part of the brain works at this time in development, but as we said, if it takes 10, 15 years to get something out into the market and for people to actually see what I've been working on from a more tangential point of view, more tangible point of view, am I really in the right place, And this is kind of why I shifted my attention towards pharmaceuticals and biotech. And so now I'm actually working for a consulting firm where I'm helping biotechnology companies and pharmaceutical companies with a variety of different problems. They're business related, but the problem, I actually am more finally in a position where I can do some impact that is a bit more visible, a bit more tangible, which is very unusual for scientists to do. I think scientists either end up in science, and I had been offered the opportunity to open my own lab, and I turned it down because I think that I like some things about science that I can find in other areas of life, kind of like you, right? The student association club that you founded, boxing was a passion of yours, but there were multiple other goals around it. You don't just like coding because you like coding or programming. You like programming because you like creation, because you like innovation. And I think for me that was the same thing with science. It's like, I love science, but what is it about science that I really love? Is it like understanding what a molecule or a protein does in their cell, or is it, do I like complex problems? Do I like solving something that people really want to know the answer to? Do I like, exchanging ideas and, sharing and communicating complex concepts in simple ways? And I found that maybe strategy consulting would be one where I could do just that. And this is why I think it's, I was invited to give a commencement speech at a university in Bolivia not long ago. And that was one of my messages. It's like, don't be afraid to reinvent yourself. If you've already, I spent 13 years in academia, and I did it. So I think,
Wow. Hey, that's so weird. I'm gonna touch base on two things that you said. First, the first thing is that in tech, I'm a software engineer. I'm a front end software engineer. And one of the reasons that I even specialize in the front end is because I can actually code something and I can see it immediately, and then build something and I can see how impactful it is to people.
yeah,
Like my first position, I wasn't only responsible for developing the application. I was responsible for designing the application. So I had to meet with like multiple different doctors and make sure that the application met their needs. And I had to make sure that the design it was easy to navigate within the application. And basically, because I had such a close relationship with those people, I can see how something that I built impacted their day-to-day life. So I completely understand that. And then engineers who are usually working on Python or they're on the back end, they're not really building anything visually. They're doing is making like computations and dealing with the math side
yeah,
of programming
exactly.
for data. And that's something I definitely. Although I got my master's in information systems, that was something that I don't really. It's not as fun. So I don't want to.
I
I
hear
don't
you.
want to go in that area. But then also you said reinventing yourself, man. That hits home with me.
Mm,
Uh, and maybe it's just I know some people are sheep and shepherd. There's no pro or con to either or. But for me, I feel like I've definitely been a different person. I've transformed and I've grown at all stages of my life, man. Going from like the hood guy to becoming the academic guy to being the athlete to being like the nerdy computer geek to being like the guitarist to I do a lot of different things,
awesome.
and I think doing these things and having these challenges for your life, it helps you. I think that keeps you young. I've seen
Absolutely.
people, yeah, I've seen people who, regardless of what their background is, if they're, this is my theory. I feel like we always have like we have an upper class and a lower class, and we kind of like will judge people based off of where they stand at. But what I think is regardless of what level you're at, I think the goal in life is to expand. So if you're here, you experience life up here. You want to experience what's going on here. Same thing if you're at the top. If you're at the bottom, you're rising. If you're in the middle, you're expanding. And I feel like when you're not, if you're here and you're not growing, or if you're here and you're not
Mm,
expanding, wherever you're not expanding, I think that actually makes you deteriorate because you're not learning. You're kind of going through the same
absolutely.
automatic process, and your brain or your body isn't like learning new ways. I guess what I'm trying to say is that, and trying to relate to what we're saying
Mm,
is that when you explore different areas in your life, it's like something happens in your brain. It's like, okay, now we have to create a new like neural pattern or
totally.
something to like, and it
Totally.
starts filling us.
Totally.
It's that energy, you know?
Yeah, for me, I love that theorem, or this theory that you have. I think for me, one that's driven me is finding this little overlap between this classic diagram that we've all heard of in terms of Venn diagrams. Like the what you love, what you're good at, and where are the opportunities. And that little overlap is where you want to be. And the problem is that opportunities in the market for jobs, they shift. Now software engineering, a computer scientist is super hot. But maybe you're not good at it, even if you love it, so you're not going to fit in there, which is my case, right? I write a little bit of code, but not much to be a computer scientist. And so I
Yeah.
think in time, as you move forward in life, like your love passions will change, the market will change, and what you become good at it will also change. And because it's so dynamic, then you actually have to remodulate yourself and where you are in this Venn diagram overlap every other year or every so often, and reposition yourself.
Yeah.
Yeah, and it's important, I
Every
think,
evolve.
to be cognizant.
What was that last?
To be cognizant of what you like and what you are, is to be aware, to be alive. To be consciously making decisions where strategic decisions for yourself, personal or professional. I think.
Yeah, none of us can escape that. It's like a walk. It's like a rock in the river.
Mm,
The rock is either going to flow with the river, or the rock is going to sit there and slowly get withered away by the ocean. You know, you're not aware of it, but I'm sure if you were to take pictures of the rock, like you were saying with the brain, you would see like the rock is changing. It's slowly dying and disintegrating because of like the waves hitting it. I want to end it with this. You kind of touched base on it a little bit. You said what you're working on, but what is the ultimate goal? What is your current goal for you right now in your current like career?
Yeah, so I guess from a scientific point of view, I'm still working on wrapping up a publication where we're studying these little nanotubes, which are tunnels that exist between cells in the brain. At least that's what had been described in vitro, which means in cells artificially grown in a plastic dish and growing in an incubator. People have heard of neurons and axons and dendrites, which is how the brain communicates. But until now, nobody has been able to understand how diseases like Alzheimer's actually propagates in the brain. I don't know if you know this, but basically, tauopathy, so basically Parkinson's disease and Huntington's disease and Alzheimer's disease are all characterized by cell death. If you look at a picture of a man that has Alzheimer's after they die, it's shrank because the cells have died. And the question then is, but why do the cells die? Because there are little proteins in those cells that are killing those cells. And so what is that?
Are those cells called prion proteins?
Yes,
Are those
yes,
proteins?
exactly. So
Okay. All right.
yeah, so
Okay.
so prion proteins or prion proteins actually are related to a disease that happened. It still happens, but it was known as the mad cow disease, and it mostly killed a lot of cows. But it can also affect humans. But there is something related to the prion proteins. And these are misfolded proteins. So basically, in your cells, you have proteins that always have a very specific shape to function. It's usually three-dimensional shape, so it's like this. And sometimes the cell makes mistakes, and instead of making a protein that looks like this, it looks like this. And normally, the cells have a machine inside of them that destroys proteins that don't look good, called the lysosome. And it's going to basically chop up these, and they're just going to reuse the materials for other things. And the problem is that if the cell doesn't destroy these things, they'll just be floating in your cell. And if it's just one, it's okay. But the problem is that it's going to continue making that mistake, and you're going to have lots of these little bad-looking proteins that are going to accumulate in the cell. And this is what is known as a plaque. And these plaques are going to kill a cell. And then you say, but it's just one cell. If I have millions of neurons, what's the problem? Nobody really knows how or why at this point. But one of the theories is that this bad protein is not just only going to, before it kills the cell, it's going to go from one cell to the other. And it's going to tell the other cell, make more of me. It's like an infection. And it's going to do this from one cell to the next, to the next, to the next, to the next.
It's going to snowball.
Yeah.
One cell talks to another cell, then two cells talk to that cell, then four cells talk to that cell, and eight cells talk. Yes.
And so then,
wow.
So the question is then how? How is one protein able to go from one cell to the other? Because we know that when in a neuron in the brain, these proteins don't really travel in the what's known as the synaptic cleft or the synapse. The synapse is the space between two neurons where the electrical current goes through neurotransmitters or gap junctions. Nobody has ever seen these little bad proteins go from one cell to the next, so there must be another way in which they go from one cell to the other. And that's where you have scientists arguing against each other, because some people say, oh, they're little vesicles, like little spaceships that come out of the cell. They're called exosomes. They're going to the other cell. But we, in our lab at Pasteur, found tubes, like tunnels, that are they just form like imagine I just start growing an arm, and I just fuse myself to you, and I just transfer this protein into your body, and in your body you just start making more of it. And that's what we saw. We literally saw this. We put cells under the microscope alive. This is in vitro, so it means that it's not a living animal. And we saw the tube grow, and we saw the little things moving into the other cell. And so this is published now. But now I'm actually trying to show this in an animal. And we finished the article, we wrote it, and now we're in the process of publishing it in a hopefully really good journal. So it's going to be a high impact factor article actually when it comes out.
Definitely, man. Send it my way.
Yeah.
I know it's just listening to you.
Yeah.
Listening to you, it's just so crazy how things are related. Essentially, what that is it's a bug. in coding, we get bugs. We have a system, a collection of services that work together, and occasionally, something won't work in the application that's supposed to work. Maybe it worked for a number of years, and you encounter these bugs. And a lot of times, you encounter bugs because certain components they know they have been deprecated. So compare that to the brain. There's something in the brain that, like, cells die. There's something there that no longer exists. And because it's dying or it doesn't work, there's things that were dependent on that component in order to function right. So because it's not dependent on it, there's bugs in your code. And if you don't address that bug, it ultimately it's going to lead to your app is going to crash eventually, and it's not going to work.
Yeah.
Now it's easy to address those bugs on code, but when it's like a human brain, it's like
It's
yeah, man,
complex.
it's a whole nother.
It's very complex.
Yes.
Yeah. And there are many challenges. There are a lot of them are technical, and again, technical from the point of view from working with cells or working with animals, all the way to having all these images and all this data and not really knowing how to extrapolate useful insights that can lead us to the conclusion or to data knowledge that could actually take us to the next level. But that's something I'm working on, and of course I have this job with a strategy firm, and so a lot of consulting now with businesses, with big large companies, and I have some personal projects as well. But I'm trying to buy a car right now, so that's something I'm pretty stoked on. Actually, when you come visit me, hopefully I'll have it by then. It's a vintage Austin Mini from the 80s, like the Mr. Bean car. Have you ever seen the show?
No, I haven't seen it, but if it's you said it's a vintage what?
Austin Mini.
Austin.
Just type in Austin Mini 1980 or 1970.
Oh wow. What made you want to get this, man? I've seen this before. I've seen it definitely in like some older movies.
I think it fits my style, and
It's a classic.
yeah, it's a classic, and it just fits Europe. The streets are not like in Salt Lake. In Salt Lake, you guys have like 12 lanes per boulevard, and here, not really. So,
Man, if you're not with somebody, I'm sure she's gonna love this. Women love like they really do.
yeah, I know. I've heard that. We'll see. But anyway, thanks so much for having me, Darren. Seriously, it's been really cool to talk. I feel like we could. There's just so much has happened between Utah and our experience. It's been almost 10 years now. So, 10? Yeah, it's been literally 10 years, I think. When did you graduate? 2012? 2013?
I graduated undergraduate. What was it? It was 2014.
2014.
And I got my master. That's when I kept trying to play football after that, but I went back and got my master's in 2016, and I graduated in 2018.
Yeah. So you really reinvented yourself with because your master's was in systems, right? You said?
Yeah, information systems, basically IT,
Wow.
information technology. So
It's incredible, man.
Yeah.
Incredible.
thanks. But yeah, man, thank you for being here. It was honestly, this is one of the best. I'll say that, but this is definitely one of the best ones. I felt like I really learned something. In this episode, I took some notes, and I'm definitely gonna, look into things a lot further.
Thank you so much.
But,
I'm really happy I joined. I'm really happy that we touched base and we got to reconnect because it had been a while even that we spoke and caught up. So this has been really cool.
It's been a minute. Do you still use passion? Do you still say that?
Passionate is just sort of in between the lines now. I don't say it as much out loud as I should. In French, people wouldn't really get it, but always.
Yeah, for the people listening, Diego, every day, passion, man, passion. That's the one thing I remember. It's like, God, man. But yeah, man, I love to see. I love where your evolution is at, and thank you for being here, man. All right.
Thanks, D. Take care. Talk to you soon.